He is currently an assistant professor of medicine at Harvard Medical School and associate bioengineer in the Division of Engineering and Medicine at the Brigham and Women's Hospital. Dr. Zhang's research is focused on innovating medical engineering technologies, including 3D bioprinting, organs on a chip, microfluidics, and bioanalysis to recreate functional tissues and their biomimetic models. In collaboration with a multidisciplinary team encompassing biomedical, mechanical, electrical, and computer engineers, as well as biologists and clinicians, his laboratory seeks to ultimately translate these cutting-edge technologies into the clinic. He's an author of more than 250 peer-reviewed papers, and his scientific contributions have been recognized by more than 40 international, national, and regional awards. The floor is yours, Shrike.
All right, thanks so much, Mehmet and Ali, for the invitation. So I'm going to talk about some of the work that I've been doing in the past few years regarding primarily 3D tissue bioprinting. So a little background to start with. So we have been really working on two different aspects. First one is tissue model engineering. Basically, the rationale for that is the conventionally used models, whether they're based on the two-dimensional static cell cultures or based on these animals, don't really translate in human system because of their differences with the human body. So that's why in the past like 10, 15 years, people are really trying to develop these high content models of the human systems, including different tissues and organs, also called organ chip systems. So with these systems, you can really model these human tissues and organs at higher content and allow really recapitulation of their functions and structures in vitro with biological and physiological relevancy. And then what's interesting is that you can also start to connect these different organ models together in a way that these organs are connected in human systems, so that you can start to also look at their interactions in vitro, right? So basically, not only organ-organ interactions, but also organ-drug interactions in this multi-organ format. So then with that configuration, you can start to really develop drugs in a better way, or if you combine those further with cells that are coming from different donors or patients, you can start to also personalized medicine, including personalized screening of the therapeutics for individual patients, depending on these scenarios as well. So, but also you can imagine all these different fabrication or whatever technologies that you use for tissue model engineering can also be adapted to the conventional tissue engineering kind of aspect, meaning that you can also start to fabricate these human tissues and organs that are functional, structured, relevant for regeneration purposes. But then these are more longer term goals, because in this case, you have to really make sure that every single tissue or organ that you make is going to be 100% functional before you actually put them back in human system, whereas in the human tissue model engineering perspective, you don't have to achieve 100% functionality, but usually 80–90% of functionality is going to be more or less sufficient. So with all those in mind, our lab has been really interested in different aspects of how we can really produce these functional humans and human tissues and organs in different ways. And these can range from biomaterials engineering to biofabrication, and in particular bioprinting, and all the way to these biomedical devices, and eventually to these bioanalysis capacities to allow us to really assemble these tissues and the organs in different ways for their different applications. So one of the examples is that we have been really trying to, for example, combine the conventional biofabrication or bioprinting methods with on-chip devices to allow production of these in vitro models that are allowed not only three-dimensionality produced by enabled by these 3D biofabrication methods, but also the dynamic cues that are enabled by these micro devices. Examples include flow, shear stresses, and also the mechanical kind of actuations. So then some examples to begin with before I transition to bioprinting on these chip devices. So these are some exemplary kind of chip devices we've been making for over the years. Examples can range from these single chamber devices that are hosting a single kind of tissue organ type in there, or having a membrane between segregating the different layers to allow endothelialization or epithelialization, or to these multi-chamber devices on a single chip device, which allows studying of tissue-tissue interactions, and also, for example, metastasis of the cells within the same kind of device, and also to these mechanically active devices to allow building in of these mechanical kind of actuations for certain tissues that are mechanically active in human system, like the blood vessels and also some of the like lung devices. But also, as mentioned, we can start to link these different organ on chip devices together in different ways to allow their interactions in vitro, right? So these are basically allowing you to really look at how these different organs are potentially interacting without being in the human system, and then how, if you put the drug molecule in there, how these different organ systems are also interaction with different drug molecules as well. So then those are some aspects on the organ chip devices, but also coming back to the bioprinting, which is the primary focus of today's talk. We have also been really interested in pretty much every single aspect of bioprinting, from hardware, software engineering to biomaterials engineering, and all the way down to the downstream applications, basically bioprinting of different organs for tissue fabrication. So examples can include, for example, multi-material bioprinting that we have been really extensively working on. The rest for that is, I mean, human tissues and organs are really complex, having multiple cell types come together, having multiple ECM types come together in the right architecture in order for the macroscale tissues to be functional. So the ability to really allow these different bioprinting methods to enable multi-material based bioprinting, that's becoming very important because you have to then have that capacity to allow fabrication of multi-cellular or multi-ECM kind of tissues for their functionality. And also we have been working extensively on these cannula tissues, basically like vessels or other types of hollow tissues that are widely present in human system, right? So we work through different technologies, including sacrificial bioprinting to these DLP-based projection kind of lithography, and all the way to these hollow tubular kind of coaxial microfluidic bioprinting methods as well. And we have been really extensively working on bioinks in addition as well. So bioinks are arguably one of the most important components for bioprinting because they not only have to allow bioprinting to happen in the first place, but also after bioprinting, they have to ensure that the cells that are embedded into into these bioprinted structures are remaining viable and functional in order for the eventual formation of the functional tissue units. So how to really design these bioinks to allow the bioprinting to happen and to secure biological functions of the bioprinted tissues, that's becoming very, very important. And we have been really interested in that kind of applications as well. And one last point is we have also been working some, not as intensively, but some efforts around minimally invasive bioprinting, basically a combination of these like bioprinters with handheld devices or things like that to allow in situ biofabrication. So I'm going to give some examples of these different aspects today to really indicate how we can use different bioprinting methods to allow fabrication of tissues and organs at high capacity. So I'm going to start with this sacrificial bioprinting that people are probably aware of very nicely already. So this is basically very conventionally used bioprinting method to create channels within a tissue construct. So what happens here is that you first deposit a fiber template with extrusion bioprinting, and then after you have that fiber template, you can start to either bioprint or cast another hydrogel matrix surrounding that fiber, and then after that, you can then start to cross-link this matrix, and once that matrix is cross-linked and stabilized, you can then selectively remove this initially bioprinted template from the hydrogel matrix, then eventually creating a hollow perfusible microchannel within the like surrounding hydrogel to allow emanation of hollow tissues like the vasculature in there. And with that, you can, of course, depending on how you print the initial templates, you can start to really pattern these hydrogel channels in different geometries, straight or like curvy channels. But then as mentioned, I mean, these channels are structurally relevant, right? Because it's hollow channel, you can start to really perfuse the channel as one would do in the in vivo situation where the blood vessels are perfusible. And if you start to really populate these microchannels with cells, these endothelial cells, you can start to also make them biologically relevant. So these are endothelial cells, and if you culture them the cells for long enough, you can actually eventually form a very nice monolayer of endothelial cells on the looming surface of these microchannels to again endow them functionality. And you can do the same thing not only on the straight channels, but also these branching channels to make endothelialized branching channels in these hydrogel devices as well. And then if you culture them for long enough, right? So because again, you have the hollow channel that's initially printed, but then you also have the hydrogel matrices that are slightly compatible in the surrounding matrix. So if you culture these endothelialized microchannel for long enough, you can actually induce also secondary sprouting of these endothelial cells into surrounding matrix to basically create this multi-scale vascular within this bioprinted hydrogel-based construct. So then not only that, you can start to also in the surrounding matrix, right? So when you try to cast that matrix, you can also embed a secondary cell type in there. For example, breast cancer cells. Now you can create a vascularized breast cancer model and look at how the endothelial cells, and in this case, lymphatic endothelial cells, are interacting with surrounding matrix embedded with breast cancer cells in a VEGF-dependent manner, as without any VEGF-C, which is a receptor for these lymphatic endothelial cells. There's very few sprouting of these endothelial cells, but you have you have higher concentration of the VEGF-C. You can now induce much better sprouting of these lymphatic endothelial cells from the original channel to the surrounding matrix, and then induce the interactions with surrounding breast cancer cells. So you can do the same things in reverse as well. So so as I mentioned here, basically what happens is that you actually had the breast cancer cells in the surrounding matrix, but now you can also create the same sacrificially bioprinted microchannels and then seed these breast cancer cells into the channels to create a model of the ductal carcinoma in situ. Basically the breast cancers that are originating from the mammary ducts, right? So if you culture the cells for long enough, you can see how the cells are gradually reaching their confluency. But then what's interesting is that once they became confluent, how the same cells were able to also start to invade from the channels into surrounding matrix, forming this invasive phenotype, as if the transition of the DCIS to ITC, right, invasive ductal carcinoma. And here's a confocal image showing the same process. Basically, not only can you see how the breast cancer cells were invading into surrounding matrix, but also how they also at the same time formed these inward aggregations, which are very similar to the in vivo configuration, right? Because in the in vivo configuration, not only the cells are going outside, but also they are proliferating within the ducts, and they eventually block the ducts at a later stage. And this is actually very similar to their in vivo configuration. And then if you look at their basement membrane deposition, they were able to also very effectively deposit collagen type 4, for example, within their growth. And if you look at their migration, looks like the invading cells were having more stronger expressions of N-cadherin than E-cadherin, indicating this potential EMT transition process for the invading cells. And back a couple of years ago, we actually also partnered with Axos, which is a space company, to sort of send some of these bioprinted mammary ductal carcinoma kind of models into a very short kind of orbital, suborbital flight, and we're able to retrieve them back from the flight, indicating that in the future, we can potentially look at how the space microenvironment, including microgravity or radiation, are potentially affecting breast cancer cell behaviors, and also how they're responding potentially differently from the to the drug molecules. All right, so then coming back to the gels itself, so we have been showing that you can very easily or conveniently create these sacrificially bioprinted microchannels within hydrogel matrix. So that's been very widely used by many different people, and that's been very instrumental over the years for creating hydrogel embedded tissue models. But then there are potentially two different aspects we're thinking about. I mean, two major challenges. One is that these hydrogel matrices can be potentially expensive depending on what type of hydrogel that you use. And second thing is that since these hydrogels are, aqueous and they degrade fast, they have to pretty much use this printed tissue models right away after bioprinting. You can't really store them for long enough, right? So with that, can you actually design some system else that can simultaneously solve these two different challenges associated with conventional sacrificially bioprinted hydrogel-based micro tissue models? So then we sort of thought about paper devices, because I mean, paper devices are very cost effective. I mean, they are very cheap. for the original materials. And then another advantage there is that once you have the paper device in the dry state, it can be stored for pretty much as long as you want, right? So then with that, can you actually combine paper devices with these sacrificial bioprinting techniques and allow that to happen? So indeed, right? So if you basically optimize the ink based off a combination of petroleum jelly and the liquid paraffin, basically wax, and directly print that ink into a matrix of bacterial cellulose, you can actually very nicely print that pattern inside this supporting matrix. And with all the optimized conditions, you can really print these patterns in 2 dimensions and 3 dimensions in whatever structure that you want. So then what happens is that once you print these structures or patterns of wax within a bacterial cellulose matrix suspension, you can then just air dry these matrices to allow eventual formation of a paper-like device that's basically made of this entangled fibrils of bacterial cellulose. But it's not only a paper device without anything. There's a paper device that's containing these sacrificially, this printed wax filaments in there, right? So then what happens is that you can, because it's wax-based, it's temperature sensitive, you can actually increase temperature to liquefy the wax and then very easily remove these wax-based structures from the paper devices to create channels, right? So you can actually create single channels or multiple channels and even channels across different layers segregated by the, again, the dense layer of the bacterial cellulose fibril, and the channels are all functional and perfusable. So then again, the rationale again to begin with, to begin the project was really the cost effectiveness, right? So if you look at the cost of these devices, and it's pretty cheap, right? So each device is only costing less than 4 cents, which is very, very cost effective as we were expecting. So then what's interesting is that another thing is that, like, it's not only cost effective, but also in the dry state, as I mentioned, you can basically store these devices for as long as you want on the shelves until when you want to use them. So when you want to use them, you can actually autoclave these devices, and that gives you 2 things. One thing is that you can sterilize the whole device with autoclave, but also at the same time, because it's water kind of a process, vapor process, you can actually also at the same time like wet the device to allow the bacterial cellulose to be become swollen into this hydrogel device, and then you can start to really put cells in there again to create these we call point of care organ chip devices by directly putting cells in there at bedside, right? So then, for example, if you can seed endothelial cells into these sacrificially bioprinted hollow channels, and then having the breast cancer cells seeded onto the surrounding matrix, you can create the vascularize the breast cancer model very rapidly on these devices, pretty much at bedside, because you can directly use patient cells that are derived from bedside for rapid assembly of the devices. And now you can start to test these drug molecules on these rapidly assembled organ chip devices for drug screening applications. All right, so then this is the first version of the paper-based devices that are sacrificially printed. So we identified one of the issues these are based off very densely entangled nanofibrils of the bacterial cellulose. So if you look at cross-sectional views, even though you can have the breast cancer cells seated onto the devices, they are pretty much on the surfaces of these devices, because again, you have very dense matrix here. The cells cannot really migrate into the interior area of the devices, meaning that you're making a pseudo 3-dimensional device, but it's not a truly 3-dimensional device for cellular interactions, right? So within that, can you actually solve this issue by treating the device with something else? And with that, can you actually bubble the device to really allow their expansion to really create these microscale pores within the layers to allow their functionality, right? So what's interesting here is that if you if you soak this device into a matrix, into a solution of sodium borohydrate, and then allow bubbling to happen, it's going to effectively expand this paper device into a 3rd dimension and create all the microscale pores that are connected within the 3-dimensionality to really allow better interactions of different cell types within the whole model that you're going to create. So here are some updates of the material itself, right? So as you may remember, so we were using these wax-based sacrificial materials as a template. But then in this case, because we're subjecting these devices into a bubbling process, which is pretty harsh, we found that if you use the same template of the wax, it's going to be deformed. So then we change the device, the template into PDMS, right? So basically, if you optimize all the conditions of the inks and also the baths, you can also very nicely print PDMS filaments into the bath and then do the same process like drying process, but eventually instead we can actually directly extract this PDMS filament with force and also create microchannels within these paper devices. But then, as mentioned, you can basically, depending on how much of the expansion process you subject these devices to, right? Depending if you can basically subject them to different concentrations of sodium borohydrate, you can subject them to the same concentration but different bubbling times. And then you can expand this device more and more as you have high concentrations or longer times, right? So without any bubbling, or original device is only about 200 micrometers in thickness. But if you bubble them for long enough and with high concentration of the sodium biohydrate, you can eventually form a device that's being expanded to a pretty thick thickness, all the way to about a few millimeters in thickness, right? And also containing all the interconnected micropores throughout the entire thickness of the device. So now, why is that important, right? So if you look at the liquid transport, not only can the liquid goes from outside to inside, but also the liquid can go from inside to outside, and this transport is becoming much easier for this expanded micropore structures than without expansion, indicating that you can also seed cells in a much better way and allow their interaction in 3 dimensions. And if you look at the cost of these devices, it's remaining pretty much the same cost, right? So the cost is only, again, less than 4 US dollar cents per device, which is pretty much similar to the previous version without necessarily having to increase the cost for these devices. And then the same procedure for cell seeding, right? So basically all the cleaving and sterilizing and then rehydrating the device. And then if you put cells in there, but what's different, you can see is without any expansion, the original device without an expansion, right? So the old version, if you have endothelial cells within a channel, the cells are pretty much staying within the channel because you have a very dense matrix of the bacterial cells surrounding this endothelial cell kind of channel. But now if you have the channel, but then the device is actually being expanded into a much thicker device with all the microscale pores that are connected, the endothelial cells are starting to really sprout into the surrounding matrix and forming this multi-scale vasculature, which is something that you cannot achieve with conventional, with the initial version of the device without any expansion. And the same thing with the whole interactions in 3 dimensions as well, right? So not only can you have endothelial cells in the channel, but also in the surrounding matrix, you can now seed breast cancer cells into the surrounding matrix. And with that configuration, you have a much better interaction of the 2 cell types in 3 dimensions, because again, you have all these expanded microscale interconnected pore structures within the whole 3-dimensional device. But in the conventional sense, right? So without any expansion, the cells are pretty much only remaining on the surfaces of these microchannels or surfaces of devices without actually being able to interact with each other in 3-dimensionality. All right, so then with our expanded devices, you can also start to look at how they're responding to different drug molecules in a time and dose-dependent manner, again, for potential drug bedside assembly and personalized therapeutic screening for the purpose. Right, so that's the first example, but also, again, we have been thinking about, this is one of the examples about how you can not only create the microchannels of the into these micro paper-based devices, but also how you can store them in a longer term on the shelves without having to use them right away after printing. So with that, can you actually have some other methods being developed to increase the shelf availability of these printed structures, because again, in the conventional sense, right? So if you print something with a bioprinter, you likely have to use them right away, again, because they're made of hydrogel devices and hydrogel materials, and they don't really last. So with that, can you actually combine a conventional bioprinting method with something that's also being used very frequently, which is cryopreservation. And now to create some technology that we term cryobioprinting to really allow not only bioprinting, but also simultaneous freezing of the constructs and then a storage on the shelf until you want to use them, right? So this gives you the capacity to really print, store in the tank, and then either use them at the later stage or transport them to somewhere else for usage at different location, right? So that's becoming very convenient with this cryo bioprinting method that we have been developing in the last few years as a new technology. So then what happens here is that you have a same printer, but the different thing is that you have a specially designed substrate that can allow sub-zero temperature to be placed on this stage. So now if you have the stage that's freezing things down, and if you start to deposit these bioinks onto the plate using extrusion bioprinting, you can start to really freeze these bioinks almost instantaneously after extrusion, right? So you can see from this movie that as you print, right, so the bioink is getting very quickly frozen, and this not only allows you to print a single layer, but you can also deposit layers on top of each other, right? So even if you have multiple layers on top, you can still slowly freeze all layers after the whole bioprinting process is done. And you can very precisely model this process with computer programming, right? So that can give you the nice idea about how the temperature and all the things are potentially affecting this freezing process. All right, so then with that, you can start to really print various patterns you want, right? So either two-dimensional, three-dimensional, and you can also combine that with multi-material bioprinting as well to create patterns of multi-material nature. But what's more interesting is that you can not only create the conventional structure that are printable potentially with the conventional kind of extrusion bioprinting, but what's interesting is you can also start to print these non-conventional structures. For example, a cylinder that has the smaller base but the larger top, which is something that's actually a little difficult to print with conventional extrusion bioprinting unless you use embedded bioprinting, basically printing into a bath, right? So, but in this case, since you're freezing down the whole filaments as you print, you can start to really print these long regular structures like smaller base, a bigger bottom top, or even this freestanding structures in vertical directions, depending on the need, right? So this doesn't need any of the supporting baths as was in the other cases or with embedded bioprinting. But you can directly print these structures with cryo bioprinting. All right, so then we look at what things eventually goal is to have cells in there. We then started to look at how you can actually create a cryopreservative bioink that allows cell behaviors or cell survival within this cryo bioprinting process, right? So first look at how DMSO is affecting ice crystal formation. As we know, DMSO is a pretty common cryoprotectant that people use all the time for cryo storage for cells. So this is actually very, conventional results, right? So if you have no DMSO in there, it's freezing. The ice crystals are growing very fast, forming this very kind of sharp ice crystals that are potentially affecting cell viability. But if you have some DMSOs in there, you can actually have a much slower ice crystal formation with much smaller crystals formed as well. And if you do this assay, basically, you can look at how quantify how the ice crystals are forming within these different formulations of bioink. And again, with more DMSO, you have much slower freezing event happening and also less ice crystals in there. And you can do the same, very similar kind of assay called splat assay, basically having a single layer of the bioink and look at how the crystals forming and the same results, right? So basically the more DMSO you have in there, the smaller ice crystals are forming within this bioink. But then what's interesting is that not only can, having DMSOs in there is going to be effective in terms of reducing cellular damages during this bioprinting process, this cryo bioprinting process. But only having DMSO there is not sufficient, right? So with control group without anything in there, of course, cells are pretty much dying after freezing. With 5% DMSO is getting better, 10% is getting even better. 15 is getting actually very high concentration, but then the cells actually started to die off again. But then if you look at 10% DMSO, for example, even if it's getting better, it's not ideal, right? Because you still see a lot of dead cells in there. So now we look at how can you further increase the cell viability during this freezing process by incorporating more other molecules, basically polysaccharides, right? So that have also been shown to be protective for the cells during this freezing process. And we did look at different effects of different polysaccharides and eventually identified as maltolase to be the optimal polysaccharide. And with, for example, 12% of the sugar plus, so we see very, very few dead cells after this freezing process, indicating that this might be the optimal condition for the cryoprotective bioink to be developed. All right, so then another thing is that, as we know, for the conventional cryopreservation, you don't want to expose the cells to DMSO for too long, right? Because again, DMSO during room temperature is going to be potentially toxic to the cells over longer term. So we also look at how long can you actually have the cells in contact with this cryobioprinting protected bioink in the presence of DMSO. And it looks like up to an hour is going to be okay, but after an hour, the cell viability is going to drop further. That's going to be very, very low to allow optimal cell conditions. So basically have to make sure that the mixture of the bioink and the cells is going to be not too long before we actually do the bioprinting process. That's about 1 hour. So we also looked at how the different layers may affect cell viability, right? So now if you print a structure that's having a pretty thick profile, looks like, I mean, even if you look at cell viability across different layers, there's not much differences across different layers in terms of their viability, indicating that during the whole freezing process, I mean, the cells are basically protected across the entire thickness of the structure that you print or cast. Also, another potential effector is the substrate temperature, right? So if you have a lower temperature of the substrate, of course, you can imagine your freezing is going to be faster, so you can potentially print the pattern in a faster way. But now we also showed that if you have the temperature to be too low, like minus 20 degrees, the cells are also going to die off, even if you have the cryoprotected bioink, right? So we eventually chose 15% as the optimal freezing temperature for the substrate for cryoprinting process. But then with all these optimized conditions, the optimal bioink conditions, optimal bioprinting conditions, you can then apply these cryobioprinting method to different cell types, right? So across different cell types spanning from fibroblasts and all the way to these primary cells, you can see that with the optimal bioink containing our sugar and also 10% DMSO, you have much, much better viability of cells after cryobioprinting than the control groups where you don't have anything in there as cryoprotective agent. Right, so then we thought, can you actually store them for different time points and still keep their viability in the bioprinted structures, right? So we did both short-term and the longer-term storage assays. So in the shorter-term cryo storage, we see that, I mean, which is 14 days, we see that the cell viability was preserved pretty nicely over this 14-day culture. 14-day preservation period. And even if you preserve them for 3 months in the liquid nitrogen condition, and if you revive the cells and cross-link and then start to culture them, the cells are also maintaining pretty high viability for both the fibroblasts and also the primary cells, right? So that's indicating that this cryobioprinting method is actually very effective in terms of both shorter-term and longer-term storage without actually having to lowering down the viability of the cells that are being printed and storage. So then we further looked at the differentiation, right? So viability may say something, but may not indicate everything the cells are experiencing. So we did further functionality assays on these MSCs. Basically, we cryo bioprinted MSCs and store them for some time, and then revert them for subsequent culture and differentiation into different lineages, osteogenesis, chondrogenesis, and also adipogenesis. And if you compare these differentiation results between the cryo bioprinted MSCs and also the control groups, which are not cryo preserved, you see that there are not much difference between the two groups, indicating that the cryo bioprinting and cryo preservation did not really affect cell functionality as well after all these processes. And not only can you create structures that are having cells in there, but also you can start to incorporate growth factors, right? So if you have a combination of endothelial cells, for example, and the VEGF together cryo bioprinted and stored, and after you write them and put them into ex vivo, ex ovo kind of CAM assay, you can see that with the combination, you have much better induction of vascularization than individual kind of components, indicating that, I mean, the cryo printed growth factors are also remaining their functionality after all these processes. All right, so then this is basic version of the cryo bioprinting. So what can you do with the technology in terms of the final applications, right? So we then actually had another variation of the cryo bioprinting, which we then term as a vertical cryo bioprinting. So we found that if you, instead of cryo bioprint these structures in a layer by layer manner, now start to really print these structures in a vertical way. You can effectively combine cryo bioprinting with the conventionally used directional freezing to allow formation of synergized anisotropy across the structures, right? So not only can you have this macro scale anisotropy with the vertically extruded fibers, but also within the fibers, you have this directional frozen ice crystals formation within the vertical direction. That's going to really create these vertically aligned microscale pores after thawing of the device, of the constructs, right? So that's allowing you to create anisotropic tissues for certain tissue engineering applications. So then we looked at how you can, how this is going to affect that, right? So if you look at cross sections, close to the bottom layer, you have smaller pores, and as you move towards the upper layer, you have larger pores created. And if you do the longitudinal cross-sectional views, you can see how the channels are actually lined along this vertical direction of the microfiber that you kind of extrude. And also there's going to be a effect of temperature, as again as the same with the previous cryo bioprinting. So with lower temperature, you can actually allow the freezing to be faster. So now you can create smaller pores. But if you have a higher temperature of the freezing plate, you have the slower freezing rate. So that's going to give you a actually larger structure of the pores that are being created. And not only can you create pores, but also you can effectively increase mechanical properties, right? Because now instead of the anisotropic, instead of isotropic matrix of the JAMA in there, you now have the vertically aligned pores across the same direction. And due to the ice crystal formation process, the polymer is also being condensed. So now if you look at the mechanical properties across both directions, you can see much higher mechanical properties in both directions, right? So than the conventional long frozen gel. So then there's also effect on the JAMA concentration at same temperature. So if you have same freezing temperature, the higher the concentration of that JAMA is, the smaller the pores are that are being created. And if you look at macro scale features of these vertically cryo bioprinted structures, right? So if you have lower temperature, then the transparency actually goes down because you have finer pores that are created. And same for the transport. If you have standard hydrogel that's not having these micro scale aligned pores, if you drop a dextrin, for example, solution on top, they are not really penetrating through because you have only the nanoscale pores, but not the microscale pores to allow transport. But now if you have the direction frozen hydrogel having all the vertically aligned pores in the vertical direction, if you drop the same dye molecule on top, it's going to very easily transport through the entire thickness of the gel, indicating that you have all these aligned pores in the vertical direction. Right, so then with this printing method, you can start to really create different structures, right? So for example, you can create arrays of these filaments that are standing vertically. You can create these filaments that are not only vertically, but also you can print them at angle, right? So all the way to like almost 0 degrees, you can also draw the fiber along that direction because it's going to be frozen within that distance. And you can also start to create these down straight, but like spiral patterns as well with vertical bioprinting. And you can have variations in there too, right? So not only can you have the single material bioprinting, you can also start to really have vertical bioprinting of multiple biomaterials. So here we basically created a multi channel but single nozzle device which have bundled channels having, let's say, 2 channels all the way to 8 channels. And if you simultaneously extrude the different biomaterials or bioinks from different channels, you can actually create these vertical filaments that are having heterogeneous structures across different filaments as well, right? So that's going to be allowing applications I'm going to talk about in the next few slides. And then not only can you do that, you can also create hollow structures. So here, if you have a coaxial nozzle, right? So basically a nozzle that's having multiple layers, you can start to also create these hollow filaments during this vertical bioprinting process, right? So it's not solid, but it's hollow filament that's having a hollow interior. And you can also fill the interior. So it's like core shell structure that's having multiple materials in different layers, and also can alternate structures, right? So having multiple kind of segments that are being printed during this cryo bioprinting process. So why is that important, right? So again, as mentioned, if you want to create models of certain tissues like skeletal muscles, because skeletal muscle cells are actually lined in one direction in the human system. With this setup, drawing the filament vertically and being able to create vertically aligned micropores in one direction, it can start to really align these skeletal muscle cells when you have cells printed during the same bioprinting process, right? So if you don't have any cryo like directional freezing process, the cells are pretty much aligned in all different directions. But now if you have the cells that are cryo bioprinted in a vertical way, having all the channels created, you can have the skeletal muscle cells aligned in one direction. That's going to be very important to model this tissue as they very nicely recapitulate the in vivo like structure. And if you quantify how the cells are behaving, right? So these are basically how the cells are aligning over days in these vertically printed cryo bioprinted structures. Day 1, not alignments. Day 3, some alignments, but by day 7, you can have a very nice alignments of cells along the same one direction of the fiber, because again, you have all the aligned pore structures along that direction. And you can compare the spreading area, I mean, between frozen and non-frozen kind of structures. I mean, if you look at frozen areas, I mean, the spreading areas, there's not much differences, but if you look at their angles, right? So the frozen samples are having much better alignment of the skeletal muscle cells in one direction than the unfrozen samples, and also the length of the microtubules, that I mean, micro like the myofibers that are forming within these structures. Right. So then another example that we give is, I mean, I talk about this multi-material capacity, right? So you can actually print 2 filaments side by side, one having the skeletal muscle cells and the other one having these endothelial cells. So now if you do that, you can over time create this musculovascular junctions having muscle cells aligned in one filament, and then the endothelial cells aligned in another filament. That's going to be allowing really nice vascularization of these skeletal muscle fibers for this engineering application. But what's more interesting is that you can actually harness how this filament is getting frozen in different sections to also create some other transitional zones of the native tissue, right? For example, we found that if you print this vertical filament, you can only print a certain thickness, right? So that's about 5-6 millimeters. And you can still sort of extrude the same filament on top, but then the section on top is not going to be frozen. That's actually getting very interesting, because now you can actually, in a single filament, create sections that are having aligned pores, and then sections that are having isotropic structures, right? So why is that important, right? So you can actually use multi-material printing to allow, for example, deposition of the skeletal muscle cells in this frozen region. But then after when it's becoming non-frozen region, you can actually have your, for example, tendon fibroblasts printed in the long frozen section. So now you can actually create the like muscle tendon kind of junction with the muscle cells aligned in the bottom section and the less aligned tendon fibroblasts within the top section, right? So indeed, if you do that, as we show here, right, having different cells in different sections. In the frozen section, you have cells that are aligned in the frozen section, but then in the unfrozen section, you have cells in there too, but then the cells are not aligning. Right, and if you put the different cell types in there, having a myoblast in the bottom section that's frozen, but having the fibroblast in the top section that's long frozen, you can start to really engineer this junction unit, having the aligned myofibers in the bottom section and then less aligned fibroblasts in the top section to really create a sort of junctional tissues for different target applications. Right, so I'm going to stop there by really thanking all the wonderful collaborators and the funding agencies and all the wonderful members of the lab who have done pretty much everything that I've talked about today. And of course, all the mentors, including Ali, who have been very helpful for I guess, all these things that I've been doing over the past years. Thank you very much.
Thank you for the fantastic talk, Shrike. Many questions came in. So which material do you use for printing and how do you develop that material filament of a 3D printer?
Yeah, so good question. So we primarily focus on JAMA, which is the gelatin derivative that can be fully cross-linked. So that's going to be allowing really a lot of the proof of concept studies because again they are cost-effective and can be used in larger scales. And we primarily focus on JAMA for most of our printing applications.
Thank you. Could you stack the paper to create larger constructs? Any issues with the mass transfer of nutrients?
Yes, I guess, that depends on the vascularization, right? So that's why we spend quite some efforts on in creating these like vascularized tissues with different bioprinting methods. Being able to really incorporate this vasculature within the tissues to allow transport of nutrients and oxygen and other things is going to be very instrumental for the survival of the cells in larger constructs.
Thank you. How do you attach your endothelial cells on the lumen? Do you use any coatings?
No, we don't use coatings. Again, going back to JAMA biomaterial, it's a very interesting material because it's naturally derived. So meaning that you can actually have cells directly attaching to the gels without having to coat the material with anything else. But in certain cases, you can also coat the material with like fibronectin or other molecules that are further promoting cell adhesion. But we haven't been seeing much issue with cell attaching onto the JAMA.
Thank you. In the simulation of bioink phase change, what type of model did you use to simulate?
So that's CFD modeling. So so basically it's a very conventional like computational fluid dynamics modeling for that purpose.
Person has a query. Is there any impact of cell morphology and cellular suborganelles and the other content like DNA and DMSO?
Well, I don't know. So we haven't done that study, but that's something that we are still continuing a lot of efforts within this cryo bioprinting kind of arena. So we definitely will be interested in seeing how the cells are potentially responding to the cryoactive agents at molecular level at some point.
Have you worked with neural cells using this method?
No, we wanted, but we didn't get a chance to do that. So we were thinking about neural cells, but we haven't just got a chance to really do that yet. Okay.
Could you comment on the scale up of these technologies and eventual translation into the clinic?
Yes, I think for now, yes, good point. It's I think one of the limitations, as I mentioned, is that you can stack things up, but then you cannot really have a much larger scale construct to be printed. So that's one of the limitations of this technology. But we're working on some potential solutions for that when we have another paper coming out in sometime in the year. So probably stay tuned for that. So I can't say much, but we have some other efforts going on around that corner.
One person says, great talk. Do you have any advice for postdocs trying to enter academia?
It's, I think that's most of the question. I mean, I guess, I mean, just follow your interest, right? So I guess whatever that interests you, I think that's going to be a good direction for you to get on to.
Okay, a few other questions. The nice cryo printing work, haven't seen it before. Can we ship for the big biomanufacturing effort happening? Can we ship cell-laden gels?
Oh, can we
Like,
ship?
Cell-laden gels, you know, you can freeze the cells and ship.
Yeah, so that's the purpose of the whole thing, right? So because I mean, in the conventional sense, if you print something, you have to pretty much use that right away. So in this cryo bioprinting method, the primary rationale is to be able to print and then freeze at the same time, and then be able to store the whole printed structures for subsequent usage, either at the on-site or you can ship them over to different sites for revival and usage. But then that's why we're also optimizing bioink to be cryoprotective, because then we do want to have cells in there during bioprinting. So we can ship print these cell-laden structures and then ship them over to different places as needed.
More questions came in. Have any primary cells been used with the freezing technology?
Yeah, so as we showed, we have been testing some primary cells like the endothelial cells and small muscle cells, and also the stem cells. And we did show that, I mean, these cells are staying alive in there, and also especially for the MSCs, they were able to also differentiate into the end lineages without showing any effects during the cryo printing process. So that's basically totally possible.
Thank you. Is volumetric paper-based vascularized tissue model implantable? What does volumetric stand for here?
Volumetric paper. Which one is that referring to?
Volumetric paper-based.
Volumetric paper-based. Yeah, so volumetric that may basically means that the structure is becoming truly three-dimensional, because as mentioned, if you don't expand the device, I mean the paper device, it's going to be having very densely entangled bacteria cells, nanofibril, that's preventing cells to infiltrate into the interior of the devices. But now if you expand the devices to three dimensions, having all these microscale pores to be built in, you can actually have very nice three-dimensionality that's built into these devices, and then allow truly three-dimensional cellular interactions within the whole thickness of the device.
Thank you. Is that implantable?
I don't think so. I mean, well, it depends on the application. For now, I mean, we're really just looking into more like organ chip or in vitro tissue model application for these paper devices. Although in certain applications like the implantable and the sensors or something, maybe that's possible, but we haven't been working on that yet.
Okay, thank you. Can you please tell about the challenges you encountered while printing the hydrogel containing nanofibrils?
Hydrogel containing nanofibers. I guess hydrogel containing nanofibers. I assume that's relating to the paper devices. I guess that's the sacrificial printing. So I guess, I mean, yeah, so again, you have to really optimize the rheological parameters and all the viscosity parameters of these different components, either the sacrificial inks or the bacteria cells baths, because then otherwise, I mean, either the ink does not form continuous filaments, or they kind of sediment into the bottom of the bath. So then you have to really spend some time to optimize all these different conditions to allow proper printing of the continuous filaments, but also when after printing, you have to allow the filaments to be staying in place in over time before you actually dry up the device.
Thank you. Any thoughts on how to remove the MSO once the tissue construct is thawed?
Yeah, so I didn't get time to talk about that. So, so it's a pretty similar process with the conventional kind of cell freezing and thawing. So, what happens is that once we bring these structures out from liquid nitrogen, we actually first UV cross-link them. So that's going to allow chemical stability, but also after that, we're going to actually subject them into immersion of the culture medium. So that's going to actually effectively exchange the DMSO out from this construct, and then you can do a few changes of the medium to really allow that to happen, and then eventually can remove pretty much all the DMS from the construct for subsequent culture and the maturation and whatever application that you want to use for them.
Thank you. Can you stack multiple printed tissue after being cryoprinted?
Yeah, that's something we're trying to look at right now. So yeah, good point. But yeah, so something's coming up for that.
For the regenerative medicine applications, I think the cell numbers limit. Have you looked into the increasing the cell numbers for proper regenerative models?
Yeah, something's also ongoing right now, so I can't say much, but we do have some efforts going on around that corner. So yeah, so we have some other additional kind of method to allow really a high cell density fabrication of tissues.
Okay, another question is, I see a lot of work in assembloids. Are you getting involved with assembloids, spheroids they assemble?
No, not really. I mean, we don't really do much about spheroids here once in a while, but not really much about that. Yeah, but maybe I think that's something potentially interesting. We just haven't been having additional effort or energy to do that yet. But yeah, definitely something interesting to consider.
Well, that's all the questions I have. Thank you for the great talk. I appreciate your time and energy. It's going at full speed.
All right, thanks a lot.